departments and disciplines. Good, they missed most of my diatribe. Across departments and disciplines, but it's also across career stages. And I've said this many times that the life and blood of a university department and school is our students. And our PhD students in particular, they spend a long time with us, several years, hopefully no more than 3.5, guys, but or 4 at the most. But and they really, that journey is just a wonderful journey. See them go from being naive to research in many cases, particularly those of us who come from a clinical background, to being world-class researchers. So this is a chance to highlight a couple of our outstanding PhD students across different disciplines, and I'll hand over to Nicola to chair the session and introduce the speakers.
So thanks, Terry. And as I just mentioned to the speakers themselves, huge respect to people at your stage at PhD studies to be willing to get up and give a presentation of their work to such an audience. I can tell you for a fact that they're great presenters, and you're going to hear about some really interesting work. And I'm really, really delighted both by the students having this initiative and by a number putting their hands up. And the 2 that you'll hear from today were selected from a number of abstracts by a team of EMCRs and our Head of Media Relations, Kerry Dunns. So we're going to start today with Jiali Tang. Jiali is in the Department of Diabetes, and she's going to speak to deletion of cell division autoantigen 1 to retard diabetic kidney disease. Jiali completed her undergraduate training and her Master of Medicine in China, and then has been here since 2020, and she's conducting this research that we're going to hear about today with Zhongling Chai here and Professor Mark Cooper. So we're really looking forward to it.
So thank you very much for the kind introduction. I'm very excited to share with you the progress of my PhD project, deletion of CD1 to retard diabetic kidney disease in DBDB diabetic mice. Nowadays, more and more people get diabetes. Diabetic kidney disease, or DKD, is one of the most common complications in diabetes. It is believed that about half of all patients with type 2 diabetes and 1/3 of patients with type 1 diabetes will develop DKD. With the dramatically increasing prevalence of diabetes worldwide, DKD has become a growing public health concern worldwide. As clinically defined, DKD is a progressive chronic kidney disease with persistent albuminuria and/or a progressive decline of kidney function in a person with diabetes. DKD is also the most common cause of end-stage renal disease, or ESRD. Once DKD has advanced to ESRD, either dialysis or kidney transplantation would be the only option for the affected individual to survive. But currently, there's no therapy available to specifically address underlying mechanisms of DKD. Current therapies can only target risk factors for DKD, such as blood glucose lowering therapies, which cannot completely prevent the development of DKD to ESRD. Therefore, new therapies are still urgently required. Our group has initially identified CD1 as an anti-proliferative protein, which is encoded by the gene TSPYL2. CD1 is also known as DEMTT. Then we discovered its role in the progression of fibrosis in DKD. The pathophysiology of DKD is complex, involving the interplay of metabolic and hemodynamic factors. Changes in these factors result in renal fibrosis and inflammation, the former being a key structural hallmark of DKD, and the latter a major key feature of DKD often observed in association with fibrosis. TGF-beta is the primary driver of tissue fibrosis in DKD, but it's hard to directly target due to its important physiological functions such as immunomodulation. Our group found that CD1 has a synergistic relationship with TGF-beta to activate TGF-beta signaling pathway by enhancing activation of SMAD3 while increasing expression of TGF-beta receptor 1, providing a potential target to reduce TGF-beta signaling, then ultimately leading to retardation of DKD. CD1 is kind of like a magnifying glass, amplifies the TGF-beta signaling from physiological level to pathological increased level. So we need to remove this magnifying glass, which means inhibition of CD1. So it is hypothesized that the deletion of CD1 can retard the development of renal fibrosis and inflammation in DBDB diabetic mice by attenuation of TGF-beta signaling. Why we chose this mouse model? As we all know, type 2 diabetes is the most common type of diabetes, accounting for about 90% of all diabetes globally. And currently, DBDB is the most widely used mouse model of type 2 diabetes, a model of leptin signaling deficiency, which can spontaneously develop obesity and insulin resistance. In this model, both early and advanced stages of DKD are developed. To verify this hypothesis, we generated a new CD1 DBDB mouse strain, producing male mouse of all 4 genotypes we needed for this project. This is the timeline of this study. Blood glucose and body weight were monitored regularly. Metabolic agent is required for each mice at 10, 15, 20 weeks of age to collect their plasma and 24-hour urine samples to evaluate kidney function in different DKD stages. Body composition were measured by echo MRI, and the experimental mice were sacrificed at the planned endpoint of this experiment to collect their blood and tissues for further analysis. So far, 16-week kidney samples have been collected completely. Real-time PCR, Western blots, and immunohistochemical staining were performed to explore both pro-fibrotic and pro-inflammatory genes and protein expressions. Morphometric analysis were also applied for these tissues to study kidney structural change. To explore the potential mechanism underlying the effects of CD1 deficiency, transcriptomic and proteomic analyses were performed on renal cortex of 16-week mice. I validate the CD1 expression in the kidney. Genetic deletion of CD1 in knockout mice was confirmed by genotyping and also real-time PCR. And we can also see that gene expression of CD1 was increased in the diabetic kidney. I've also confirmed the absence of CD1 by Western blot in testes. No band was seen in CD1 knockout groups. Then let's see the blood glucose analysis. As expected, all the DBDB mice have a much higher level of blood glucose than DBH mice, but there's no significant difference between CD1 wild type and CD1 knockout groups for both blood glucose and HbA1c, suggesting that CD1 deficiency doesn't significantly affect glycemic control in DBDB mice. Then body weight. Again, all the DBDB mice were obese and all the DBH mice were lean as shown in this figure. And we can also see that DBDB CD1 knockout mice have a lower body weight than DBDB CD1 wild type mice, but such difference is not observed in control groups. Analyzing the data at 20 weeks of age, we can see the difference between CD1 wild type and CD1 knockout diabetic groups is statistically significant, and same as the results for BMI, suggesting that CD1 deficiency can reduce body weight in diabetic mice. Consistent with the body weight finding, the results for body composition measured by echo MRI show that both lean mass and fat mass was decreased in CD1 knockout diabetic mice. I've also calculated the percentages for lean mass and fat mass and found that deletion of CD1 increased lean mass percentages from 47 to 49%, but reduced fat mass percentages from 53 to 51% in obese diabetic mice. But CD1 deficiency has no significant effect on lipid levels in 16-week DBDB mice. Now I'll show you the metabolic aging data that I've collected so far. As expected, there's an increase in daily water and food intake as well as urine output in diabetic mice. But there's no significant difference between CD1 wild type and CD1 knockout groups at 3 time points, except for water intake at 10 weeks of age. But such difference isn't observed at the next 2 time points. Continuing with the metabolic aging data, increased urinary albumin excretion is observed in both CD1 wild type diabetic mice and CD1 knockout diabetic mice. At 10 weeks of age, there's no difference between these two groups. 6 weeks later, at 16 weeks of age, we can see the urinary albumin was further elevated in CD1 wild type mice, but reduced in CD1 knockout mice. Furthermore, CD1 also tended to increase the creatinine clearance in diabetic mice, indicating improved kidney filtration. All these results suggest that CD1 deficiency may improve kidney function in diabetic mice. As I mentioned before, CD1 has a synergistic relationship with TGF-beta to activate TGF-beta signaling pathway. Inhibition or deletion of CD1 would attenuate TGF-beta signaling. Indeed, genetic deletion of CD1 in diabetic mice results in a significant decrease in these TGF-beta related gene expressions, including TGF-beta and TGF-beta receptors, even in non-diabetic group. CD1 deficiency decreased TGF-beta 1, TGF-beta 2, and TGF-beta type 1 receptor expression in CD1 knockout control groups. Furthermore, IgG staining shows increased expression levels of phosphoSMAD3 in CD1 wild-type diabetic mice, which were attenuated in CD1 knockout diabetic mice. All these findings highlight the regulatory role of CD1 in modulating TGF-beta signaling pathway. So, the absence or inhibition of CD1 is expected to attenuate renal fibrosis and inflammation by inhibiting TGF-beta signaling. Indeed, CD1 deficiency in diabetic mice significantly attenuated this increased fibrotic gene expression, including collagen, fibronectin, alpha-SMA, MMP2, and MMP9. The Western blot's results show the consistent trend for alpha-SMA, in line with the real-time PCR results. Consistent with the reduction of pro-fibrotic gene expression, increased staining for fibrotic area in diabetic CD1 wild-type mice was reduced in diabetic knockout mice. Furthermore, the kidney structural alterations linked to diabetes, indicated by elevated glomerular sclerotic index, were ameliorated in CD1 knockout diabetic mice. I've also looked at the inflammatory gene expression in kidney. We can see that CD1 deficiency results in a significant decrease in all these increased pro-inflammatory gene expression in diabetic mice. So far, we have partially verified our hypothesis, but we are still curious about if there are any other mechanisms underlying the CD1 deficiency effect. So we performed both bulk mRNA sequencing and proteomic analysis on 16-week renal cortex tissue. I identified differentially expressed genes and proteins which were altered by diabetes but resolved by CD1 knockout groups. Then I performed pathway enrichment analysis for these proteins and genes based on 3 databases, KGG, Reactome, and Wikipathway, and identified many pathways. I found some pathways are both enriched in mRNA-seq and proteomic analysis, including TGF-beta SMAD and TGF-beta non-SMAD signaling, also complement cascade, and cellular senescence. Translation is the most enriched pathway in proteomic analysis, suggesting a potential mechanism underlying CD1 deficiency effects. NF-kappa B signaling is another relevant signaling pathway which enriched. Furthermore, the top 10 hub genes were determined based on the string database. All these proteins are ribosomal proteins. Ribosome is an essential cellular machinery that is responsible for translating mRNA into proteins. They are made up of 2 parts, a large and a small subunit. Each subunit consists of ribosomal RNA and ribosomal proteins. These ribosomal proteins are clearly closely related to ribosome pathway and cytoplasmic translation in biological processes, and they are notably enriched in large ribosomal subunits in terms of cellular components. All these ribosomal proteins are mostly enriched in RNA binding and mRNA binding in terms of molecular function. Interestingly, more than 10 years ago, our group performed E2 hybrid screening to identify proteins interacting with CD1, and we found that many ribosomal proteins as shown in this table can bind with CD1. So taken together, CD1 may influence key translation processes by interacting with ribosomal proteins or RNA, thereby affecting those signaling pathways. According to all these transcriptomic and proteomic results, we speculate that targeting CD1 can modulate all these signaling pathways by affecting ribosomal function, thereby attenuating renal fibrosis and inflammation in DKD. I've already talked about TGF-beta SMAD signaling before. For TGF-beta non-SMAD signaling pathway, MAPK-ERK. The proteomic results show that MAP2K1 and MAP2K2 is elevated in CD1 wild-type diabetic mice, but reduced in CD1 knockout diabetic mice. These 2 proteins are upstream kinase of MAPK or ERK in the MAPK cascade, which can facilitate renal fibrosis. So I checked the expression of activated ERK or phospho-ERK by staining and Western blots. Both results are consistent with the proteomic data. Furthermore, all these findings were also supported by our group's previous in vitro data. In mouse primary kidney cells, TGF-beta failed to elevate the expression of phospho-ERK in CD1 knockout groups. Then for Wnt beta-catenin signaling, Wnt ligands induce beta-catenin stabilization can enable beta-catenin to complex with SMAD proteins and enhance transcription of fibrotic molecules. I did Western blots for beta-catenin on few 16-week kidney samples. We can see that beta-catenin is increased in diabetic wild-type mice, but decreased in knockout mice. So to summarize, CD1 deficiency is shown to reduce both inflammatory and profibrotic gene and protein expressions in 16-week diabetic kidney. This occurs by inhibiting TGF-beta SMAC and non-SMAC signaling pathway. This occurred without affecting blood glucose and lipid levels, but it did result in a slight decrease in body weight in 20-week mice. And I will continue to verify the other mechanisms, including NF-kappa B, complement cascades, and cellular senescence. Lastly, I'd like to acknowledge my supervisor and the Department of Diabetes for their great support and encouragement. That's all. Thank you.
Any questions from the audience? Oh, Emma, thank you. You have the microphone. Great. And maybe I'll just get Emma start. You started at the very beginning with showing that CD1A was upregulated in diabetes.
Yeah.
Do you know what the regulations are? What's driving that? Is that related to the inflammation?
You mean the regulators for CD1? Oh, actually, we are not clear about this. Yeah, we just see this phenomenon.
Okay.
Yeah, CD1 was increased in diabetic kidney.
And related to that,
Yes.
of course, fibrosis is a problem and other diseases. Do you know if CD1A is upregulated in pulmonary fibrosis or liver disease?
Liver disease? Yeah, I've read that those like literature that TGF-beta signaling is also increased in other fibrotic diseases. But for CD1, we haven't explored the expression level. Yes.
Right, certainly TGF-beta. So
Yes. Yes, but we can try.
Right. Jerry?
Really nice work. And have you thought about, the group thought about how this mechanism potentially patient with diabetes to prevent diabetic kidney disease? Is there ways of targeting it that could be translatable to patients?
Thanks for your question. Actually, our group has previously generated a peptide inhibitor for CD1 named as CHA061. It's already been validated in activin-induced diabetic mouse model, but we're still optimizing this inhibitor. So maybe in future it deserves to try this new inhibitor in type 2 diabetic mouse model, so we can translate these findings to clinical.
Can we check if there's questions on them?
Sure. Stop sharing the screen now.
So, any questions online? Just unmute and ask. Otherwise, thank you very much. JD, we'll move on to the next speaker. Yeah. Maren knows what it's doing. So whilst Maren opens up her talk, it's my pleasure to introduce her. So Maren is a clinical fellow and at the Alfred in the epilepsy team and a PhD student in the iBrain lab, which is led by Professor Nina, and the main supervisor is Lucy Vivash. Maren, for those of you who haven't heard her speak in the recent 3MT competition, is working on using imaging as a predictive power for epilepsy surgery, and we're really looking forward to hearing about the outcomes of that work now.
Thank you, and thank you very much for the opportunity to speak to you about some of the research that I've been doing, and I'm hoping today to take you through a few of the projects I've been working on, some that are finished and some that are ongoing. So just to start off with, at least 1/3 of patients who live with epilepsy have seizures that can't be controlled by medications alone, and we call this drug resistance. And we know that epilepsy surgery is the best treatment option for people with drug-resistant epilepsy, as it offers the best chance of seizure freedom compared to continuing with medication alone. And when we refer patients for epilepsy surgery, our goal is to induce remission of seizures, but also to simultaneously preserve eloquent brain cortex and cognitive function. And this goal is only achievable if we can localize the epileptogenic zone, which is the area of the brain from which seizures arise, and if this is amenable to safe resection. And unfortunately, despite all of our best efforts and rigorous selection processes, not every patient who has epilepsy surgery becomes seizure-free. And although it depends on the individual patient characteristics as to the odds of them becoming seizure-free, on average, it's approximately 60 to 70% of patients. In this study published in 2022 by Alma Vassi and colleagues, they looked at meta-analysis data for various different pre-surgical and surgical factors that were associated with outcome in epilepsy surgery. And here in red, I've just highlighted the ones that were consistently prognostic. And I'll draw your attention to the focal imaging abnormalities, of which MRI and SysCom were both consistently prognostic. Also, they found that increasing degrees of multimodal concordance, as well as surgical factors such as the location of the resection and whether or not a complete excision of the lesion or epileptogenic zone was undertaken, were also prognostic. You can see here that FDG PET, which is a form of nuclear medicine brain imaging, that looks at the brain's metabolism of glucose, was of uncertain prognostic value, and this was because there were mixed data in the previous meta-analyses. But just regarding FDG PET, we know that low glucose metabolism or hypometabolism is a common feature on the brain PET scans of patients with epilepsy, drug-resistant epilepsy. And it's commonly used in the evaluation of patients prior to epilepsy surgery. But its use around the world is centre-to-centre dependent. And some centres, like here at the Alfred Hospital, we perform a PET scan for every patient who's being evaluated for epilepsy surgery. But at other centres, it's used in a more limited fashion for patients with either MRI negative epilepsy or where the other pre-surgical data is discordant. And there are no international consensus guidelines on how we should evaluate patients prior to epilepsy surgery. But some expert opinion articles have suggested that PET should be used only in these limited situations. But we also know that PET is cost effective and it's non-invasive. And so we think probably the main limitation to its broader uptake for use in epilepsy surgery is the evidence for its usefulness. And there's a rich history of previous research looking at PET and epilepsy surgery from members of the Department of Neuroscience. And I've just here highlighted a couple of articles from members of our lab group who have looked at various things. The first one looking at the distribution of PET hypometabolism and outcome, which found that the presence of contralateral hypometabolism and a lower volume of the hypometabolism zone resected were both associated with worse outcomes following epilepsy surgery. And then these features, these extracted features, were used in a machine learning model to predict epilepsy surgery outcome. And it was found that these could be used with AUCs of up to 0.8. And importantly, we also found that, or previous work also found that the models that incorporated surgical data performed better than the models that didn't. The natural progression of this work is that we wanted to use this information, the PET scan, the MRI and the resection region, the surgical data, and incorporate them into more complex forms of machine learning to predict surgical outcome. But firstly, I wanted to look at PET because there was some uncertainty regarding its usefulness in previous meta-analyses. So the first study I've been doing in my PhD was looking at the association of localizing hypometabolism on PET scan and outcome following epilepsy surgery in the form of a meta-analysis. And secondarily, we looked at this question of the distribution of hypometabolism, as well as whether it mattered if an epileptogenic lesion was detected on MRI, as is proposed by those previous expert opinion articles. Also the role of multimodal concordance and whether it mattered where the location of the resection was. So we included studies that evaluated patients with PET prior to epilepsy surgery that had at least 10 participants and which reported a surgical outcome at 12 months. And of the 12,820 potentially eligible studies identified at screening, we ultimately included 98. We defined localizing hypometabolism as concordant with the resection zone, which is a proxy measure of the epileptogenic zone. And we defined a favourable outcome as consistent with Engel class 1 or ILE classes 1 to 2. And broadly, these encompass complete seizure freedom and non-disabling focal aware seizures. So you can see that what we found was that the odds of achieving a favourable outcome following epilepsy surgery for patients in whom there was localizing FDG-PET hypometabolism were 2.68, and this was significant with a confidence interval of 2.08 to 3.45. You can also see that we grouped the studies based on the method of analysis, and in our stratified meta-analysis, looking at this, we found that this results were significant for visual analysis and for more complex methods of semi-quantitative and quantitative analysis. Regarding the distribution of hypometabolism, we also found that focal PET hypometabolism was associated with better outcomes in epilepsy surgery than both normal FDG PET and diffuse hypometabolism. And interestingly, we found that localizing hypometabolism on PET scan was associated with better outcomes in patients who both had a positive MRI and a negative MRI, suggesting that a PET is useful for all patients regardless of their MRI result, in contrast to those previous articles. In terms of the resection location, unsurprisingly, the majority of studies focused on temporal lobe epilepsy, and in this subgroup, there was a significant association with PET hypometabolism. But in the frontal group, you can see there were fewer studies, fewer participants, and this result was not significant. However, it was close, it just crossed one by a small margin, so it's possible that this is underpowered. And then for the question of multimodal concordance, we found that concordance of EEG, PET, MRI and PET, and triple concordance with all three were associated with better outcomes following epilepsy surgery. And this aligns with the results of previous studies, as I mentioned, that indicate that increasing degree of concordance, multimodal concordance, is associated with better outcomes following surgery. So overall, from the study, we conclude that the use of PET is useful for all patients who are being evaluated for resective epilepsy surgery. And we do think that it can help to improve patient selection for surgery, but it can also provide more information to help counsel patients about the likely outcomes from surgery. And it may delineate patients in whom it would be better to utilize other investigations such as stereotactic EEG prior to surgery. So moving on from looking at the evidence for PET, the next thing is that in my upcoming machine learning analysis, we want to be able to incorporate resection region. And to do that, we need to be able to segment the resection region to use it as an input. And the gold standard for brain region of interest segmentation on MRI is manual segmentation, but this is labor intensive, it's time consuming, and in our machine learning analysis, we wanted to be able to include 1,000 patients, and that would be very time consuming. So we looked at publicly available automated segmentation tools and wanted to compare the performance of those tools in our cohort. And one of the other factors is that the tools that are available, which there are 4 publicly available tools, have primarily been optimized for use in temporal epilepsy surgery. So we also wanted to see how they performed in extratemporal surgery. So we included 50 subjects who had had epilepsy surgery at the Alfred Hospital and the Royal Melbourne Hospital, and 30 of these were temporal, 20 were extratemporal. And you can see here a sped up screen recording of me manually segmenting the resection cavity, and it's about 10 times the speed which it actually takes, which is probably about on average 30 minutes per subject. So I did this for all of the 50 subjects, and then also ran each of these 4 automated segmentation pipelines. So EpicChop, which was developed by Dr. Ben Sinclair, who is one of my supervisors and from our lab, as well as ReceptBowl, which was developed by the University of Campinas, both use STM 12, which is a program within MATLAB. And essentially what this program does, or these algorithms do, is generate a difference map between the pre and post-operative MRI. The other 2 algorithms there were ResEg and deep resection, and these use deep learning to segment the resection region. We subsequently compared the manual segmentation to the automated segmentation using the dissimilarity coefficient. And here you can see some examples of various different types of resections and how each of the algorithms performed. So in row 1, this is a larger anterior temporal lobectomy, and you can see that most of the models perform pretty well compared to the manual segmentation. However, in the smaller resections and in the extratemporal resections, there were some inaccuracies and some where the models missed the resections entirely. So overall, none of the algorithms were able to identify every resection cavity. However, as you can see, the SPM-based tools did perform better than the deep learning tools in our cohort, and EpicChop had the highest median DICE score of 0.71. When we looked at the temporal group alone, again, the detection rates were higher in the SPM tools than the deep learning tools. However, the most accurate was ResEg. And in the extratemporal cohort, the SPM tools were a lot more able to detect a lot more of the resection cavities than the deep learning tools. So from this, we found that automated pipelines can reduce the time to segment the resection cavity. And in our cohort of mixed temporal and extratemporal patients, the SPM-based tools did perform better than the deep learning tools, but none of these pipelines was able to identify every resection cavity. So when we're using this, we do need to use quality control. So back to have this outline, having looked at PET and also how to segment the resection cavity. The next project in my PhD is looking at developing this deep learning tool that uses these 3 inputs: the preoperative MRI, the preoperative FDG PET scan, and the resection region to predict outcome following epilepsy surgery. And really the goal of this study is to move beyond these less complex forms of machine learning, which had been used in that paper by Ben Sinclair previously, into more complex forms of machine learning in the form of a convolutional neural network. So in this, we are including patients who have had epilepsy surgery for drug-resistant focal epilepsy since the 1st of January 2000, who are more than 16 years old and have the available data, being the pre and post MRI, PET scan, and the outcome at 12 months. And ideally, we'd like to include 1,000 subjects. 1,000 subjects is a lot of subjects for epilepsy surgery, and so we have been partnering with some Australian and international collaborators. And as you can see, we have also partnered with Enigma Epilepsy, which is an international collaboration for epilepsy neuroimaging research. We have groups from Melbourne and Sydney, as well as North America, South America, and Europe, who are providing data for our study. From these sites, we're projected to have 963 patients worth of data, but so far we've collected complete data sets on 259. As I mentioned previously, so for the data pre-processing, we are generating the resection region by using EPIC-CHOP, and this does have times where it's less accurate, either incompletely segmenting the resection cavity or sometimes missing it entirely. And some examples of where EPIC-CHOP has gone wrong I've put here on this image. And so some of what I'm doing at the moment is working on the data quality control. So where the EPIC-CHOP has failed, we are manually adjusting the segmentation. So my role in this project is mainly on the clinical side, but the model itself is being developed by Dr. Ben Sinclair, and we're calling it PROPHET tentatively, which is the pre-trained ResNet for surgical outcome prediction using PET, hypermetabolism and excised tissue. Bit of a mouthful, but PROPHET's probably easier to go with. And so far, the preliminary versions of PROPHET that have been developed have been on very small subsets of the data. We've got a lot of data coming in at the moment, and so the early work was mainly on data from the Alfred, which was a small subset. And unfortunately, in those early ones so far, the network isn't learning and it's classifying all patients as seizure-free. However, we do think that this can be overcome, and some of the steps that we're going to use include using more data, obviously, and we would also we're going to attempt to use less complex models with fewer parameters to try and reduce the problem of overfitting, as well as try and grade metamask rather than the entire T1 MRI for the preoperative MRI. And then in terms of future work for this study, not started yet, but planned is a prospective validation of PROPHET. I've mentioned already, but this tool is being trained on the actual resection region of patients who've already had surgery, but to be clinically useful, it needs to be able to be used in a pre-surgical way. And so in order to do that, the tool will hopefully be able to be used with the intended resection region. And so the plan is to prospectively validate the tool using by doing a prospective cohort study and manually annotating the intended resection region prior to surgery, and then using that for validation of the tool. So this work would not have been possible without the support and involvement of many collaborators, but I would like to particularly thank my supervisor team, and a special thanks to Dr. Lucy Vivash and Dr. Ben Sinclair. And I'll leave you with this image I created for my 3-minute thesis. And I hope you welcome any questions. Thank you.
Parents and Passport staff. Any questions? So maybe a game of start with a non-neuroscientist, possibly very naive question. I expected at the very beginning when you introduced the glucose imaging studies that an area of, I understand, hyper-excitability and hypermetabolism. Is it known why it's hypermetabolism signature?
Essentially we think that hypermetabolism reflects dysfunctional brain cortex, and that's kind of what we see with a lot of patients who have epilepsy. That's the area from which seizures arise is dysfunctional, but there are times where hypermetabolism can be present on PET scans in for patients with epilepsy, particular pathologies can be associated with it, such as vocal cortical dysplasia, some of them, but in general, obviously low glucose metabolism, hypometabolism, and exactly what causes that, I'm not sure. I don't know whether we know that exactly. Yeah.
Derek.
It's great work, Mary. How about that? Wonderful. Now, this is partly a philosophical question and partly a guiding fixer. So, the information that you're largely feeding into the algorithm is the sort of information that we feed into our non-artificial intelligence epilepsy surgical program decision-making all the time. And we still only get 60 to 80% prediction rate in seizure freedom. Do you think fundamentally all this artificial intelligence can actually improve on that, given it's still using essentially the same information? And yeah, there can all be all sorts of advantages about standardising, automating, rolling out in many different countries. But do you think it's still not, we're missing something in terms of what the determinants of those 20 to 40% of patients that don't become seizure free? Or do you think there is some real clues in the PET hypermetabolism pattern you're bringing in that might elevate that outcome prediction risk outcome?
Yeah, I mean, that's a good question. And the nice thing about a philosophical question is I can say, I don't know, but I think that a couple of points. I think some of the main reasons for surgical failure is we want to make sure that we're doing the right surgery for the right patient. So no one surgery is necessarily right for every single patient. And there may be factors for an individual patient that can help us work out whether or not that individual patient is going to become seizure-free. And when we look at the data that's been previously looked at with imaging data, MRI, PET, other things, these are all population studies looking at population-wide associations. But what our tool will do, hopefully, if it works, is be able to predict for an individual what that individual's chance of seizure freedom is. I guess the thing about deep learning is that we're not using features that we extract from the data that we think are important, like, you know, the presence of hippocampal sclerosis on MRI or, you know, focal hypometabolism on PET scan, but we're feeding in the full data and allowing the machine to learn from that data. And so there may be different features in that data set that help it to predict outcome. And so, yes, fundamentally, I do believe that we can do better, and I do believe that our model can predict better, and that's why we're doing it. But that remains to be seen, I suppose.
And maybe on more curiosity, but
yes.
Deletions change size over time, and if yes, is that likely to impact on we're going to get from
so in terms of MRI lesions, it depends on what the lesion is, and we do see, like when we do serial MRI, that sometimes the appearance of changes on MRI does change, and if it's a particular thing like a, you know, a tumor, maybe that will change, but if it's a malformation or development, that really won't change that much.
I was thinking specifically that.
Yeah, so when you mean on MRI or on
by whatever you need to look at?
Yeah, so I mean the like the epileptogenic lesions on MRI can include tumors, can include FCDs, can include hippocampal sclerosis, and so it depends on what it is as to whether that will change over time. In terms of PET, I think that most of the time the PET findings don't change. I think that there have been studies that have looked at that, although I'm not sure whether that's something that's an actual effect, or whether it's just that it hasn't been looked at properly yet. Yeah.
Very cool. We'll just check. if anyone has a question online, just put your hand up or put something in the chat. Thank you. All good? Well, I think there's lunch. Jiali, what do you know?
So yes, it's one of the attractions of coming to this face to face. You actually get fed intellectually and physically.
So, thank you. Let's get the
Thank you, Nigel and Camilla, for organising and selecting two fabulous presentations. So thank you, Geraldine and Maren, for that. Just a little bit of a plug. Really important for the school. We have the dean and the deputy dean for research and deputy dean for education. The faculty heavyweights all coming to our precinct on Thursday morning, and it would be really good if we had as many from across the career spectrum and departments come to support that and listen to what they have to say. Mary circulated several times the invitation. You just click and reply that you'll be coming, and then please come. Apparently at the Monash Medical Centre, 150 people replied they would come, and about 50 turned up, so we need to do better than that. And there will be lunch then too, or something.